Let us learn the diagram of TS of leaf today. So the diagram label is TS of leaf, that is transfer section of a leaf. This is most important for the SSC students, the state syllabus students. So what you have to do is, whenever you draw a diagram, first learn all the labels properly. First you draw it once or twice, learn all the labels. Based on the labeling only, we can draw any diagram easily. So when we think about the TS of the leaf, you have first what is called as the cuticle layer on the top, like this. We have cuticle layer on the top, and also at the bottom you can represent it like this. So this is the cuticle layer what we are drawing. Then we have the layer of upper epidermal cells. What are these? These are the upper epidermal cells. So followed by that we have the lower epidermal cells onto the lower side. So both sides you have to draw the epidermal cells, which will be the outermost cells in the plant. So you will draw the upper epidermis, lower epidermis. On the top of that you are drawing the cuticle, and in the lower part we have the stomata. So what we can do, you can draw the guard cells like this, and then give a space in between, which will be your stomata. This is the stomata, and what we have drawn till now, this is the upper epidermis, and then we have is the cuticle is the first structure what we have drawn. So this is the cuticle part. Now coming to the other parts, so first what you have to remember is, you have to draw a skeletal structure wherein you will fill all the details very easily. So let us put a skeletal structure in the middle like this, which will be for your xylem and phloem. Some part we are just leaving to put the xylem and phloem. Then we will fill the other space with the palisade cells or the mesophyll cells, which are the cells in the leaf. So you will represent them in the form of long balloons. You know, elongated balloons will be there in the market. So you can draw them like that, long balloons like you draw. The first layer should be drawn very closely to each other. The cells should be close like this. And when you are going for the second layer, see that in between two cells you draw one cell like that. So like this you can go on filling your structure like that with all the palisade cells. Then coming to the third layer, when you are drawing, see that there is some space between the cells, which will be depicting the spongy parenchyma region. So here, if I have to draw the spongy parenchyma, so I will draw the cells far away from each other, creating some space in between. So this space basically would be helping us to represent the spongy parenchyma region. So you can draw like that, and then we have is the next layer of the palisade cells. You can draw like that and complete the diagram. So wherever the space is left over like that, you draw the palisade cells. So all these are the mesophyll cells or the palisade cells of the plant. Then whatever we have left the place in the middle, that part we will fill. So first we will draw what are called the bundle sheath cells, which will be present around like this. So you can just cover the area like that with the bundle sheath cells. These are called the bundle sheath cells. What are those? These are the bundle sheath cells. So you can refer to the video wherein I have described about the TS of leaf. So there you can see these are the bundle sheath cells. Then inside this one, the space what we have left, there we have what is the xylem and phloem, the vascular bundles. So xylem, usually you can represent it something rigid structures, rigid cells like this, dark colored. These are all the xylem cells that you can represent as. And then coming to the phloem part, we can represent the phloem. You can just represent the phloem using another color so that it will be clear for you. So the phloem part will be like this, the cells will be like that. So like this completes the diagram. So this one is the phloem, what we have drawn, and this is the xylem. The blue one are the xylem. And what you have to remember is, we have to put the chloroplast inside the mesophyll cells. So the dots, whatever you put inside, these are all the chloroplasts. So remember that you put dots inside all the mesophyll cells and fill them without fail, so that they will be representing the chloroplasts. So you fill afterwards, and these you can write them as the chloroplasts. So we have the cuticle, we have the upper epidermis, we have the chloroplast, we have the mesophyll cells. These are the mesophyll cells. So the mesophyll or the palisade cells we have. So we have the spongy parenchyma region. So this region is the spongy parenchyma region. And then we have the lower epidermis and stomata. So like this you can easily draw the diagram of TS of leaf. Hope you followed. So do remember the label to draw the diagram. So in the coming up short, I will be telling you how to draw the TS of the chloroplast as well as the human digestive system. Thank you.